Well, hello and welcome to everyone joining us for this X-Talks webinar. Today's talk is entitled Strategies to Reduce Risk in Multi-Site Assay Cross-Validation. My name is Corey Stanton and I'll be your X-Talks host for this event. Today's presentation will run for approximately 60 minutes or so, and the webinar includes an interactive Q&A session at the end. To make the most of this session, feel free to submit questions and comments for our speakers throughout the presentation, and we'll try to attend to as many as possible. To do so, you can use the questions chat box, which is located in the control panel on the right-hand side of your screen. And if you require any assistance, please don't hesitate to contact me by sending a message using this very same chat box. Please note that all audience members are in listen-only mode and that this event will be recorded and made available for streaming on xtalks.com. And now let's take a moment to meet our panelists for today's presentation. One of our speakers today is Dr. Lynn Kamen. Dr. Kamen is a scientific officer, executive director at bioanalytics, and she received her PhD in immunology at the University of Michigan and completed a postdoctoral fellowship in immunology at the University of California, San Francisco. Dr. Kamen is an accomplished scientific leader with over 15 years of experience in drug development and deep technical expertise across large molecules, small molecules, and ATMPs. Her experience spans multiple therapeutic areas, including oncology, cancer immunotherapy, immunology, ophthalmology, and neurology. Welcome, Lynn. And also joining us today is Jessica Bridges-Weaver. Jessica is an accomplished scientific leader with over 2 decades of extensive experience spanning the entire CMC lifecycle. From upstream and downstream process development to complex manufacturing and analytical development, Jessica has built a remarkable career. Currently serving as Scientific Officer CMC at bioanalytics, she bridges the gap between advanced scientific operations and commercial success, bringing a wealth of strategic and hands-on expertise to the table. Jessica was also recently recognized with the 2026 Triangle Business Journal's Women in Business Award and is deeply passionate about advancing biomanufacturing, driving robust analytical strategies, and mentoring within the STEM community. Welcome, Jessica. I'm also very pleased to welcome Dr. Pavan Prathapati. Dr. Prathapati is a Manager 2 at bioanalytics, specializing in LC-MS-based bioanalytical method development, validation, and regulated sample analysis. He supports complex drug development programs with expertise in assay optimization, troubleshooting, and analytical strategy across multiple therapeutic modalities. Dr. Prathapati brings extensive experience applying mass spectrometry technologies to deliver high-quality, scientifically rigorous data that supports regulatory and development objectives across all phases of drug development. Welcome, Pavan. And also joining us today is Dr. Brian M. Gulick. Dr. Gulick serves as a Director of Bioanalytical Operations at bioanalytics, where he oversees the molecular programs in Durham. He earned a PhD in biochemistry from the University of Alabama in Tuscaloosa, while researching the role of chromium in type 2 diabetes. He has worked in analytical and bioanalytical development throughout his career, leading assay development and validation of small and large molecular entities. Dr. Gulick has been leading and managing PCR-based workflows for the past half-decade plus, leading efforts to optimize quality and efficiencies and improve productivity. Welcome, Brian. I'm also happy to introduce Michael Mullen. Michael is a Scientist 1 at bioanalytics, supporting the bioanalytical studies through regulated laboratory operations and scientific execution. He contributes to assay implementation, sample analysis, and data generation across drug development programs, helping ensure high-quality analytical performance and operational consistency. Michael brings hands-on laboratory experience and a collaborative approach to supporting complex bioanalytical workflows within regulated environments. Welcome, Michael. And rounding out the panel today is Todd Lester. Todd is a seasoned biotechnology and pharmaceutical project management professional at bioanalytics with extensive experience leading complex bioanalytical studies across drug development programs. He oversees technical strategy, study execution, data interpretation, and reporting, helping ensure scientific rigor and regulatory compliance. Todd brings broad GXP expertise and deep knowledge of FDA and EMA expectations, particularly in immunogenicity assessment strategy and bioanalytical program management. And welcome, Todd. And now, without any further ado, I'd like to hand things over to Lynn Kamen to kick off the presentation by telling you a little bit more about our X-Talks partner for this event, bioanalytics. Lynn, over to you.
Thank you so much, Corey. Really appreciate it. Let's go forward to the next slide. So today's presentation is titled Navigating Assay Cross-Validation, Key Strategies and Considerations. Next slide. And in today's talk, I will first give a quick introduction to bioanalytics, who we are, what we do. I will then provide a quick overview of cross-validation, and then we're going to get into the meat of the presentation, where we talk about assay transfer and cross-validation case studies across a variety of assay platforms, including PK, PCR, flow, and CMC. We will finish with some guidance on ADA cross-validation for anti-drug antibody assays as well. Next slide. So we are a global CRO with headquarters in Durham, North Carolina, sites in Boston, San Diego, Hamburg, Germany, and Australia. We also have a partner lab in Beijing, China. So we really can support any global study that you have need of. Next slide. We support every phase, every modality, from early stage drug discovery through post-marketing commitments, and we support all drug modalities. A snapshot below is just a small sample of the different studies that we've supported over the past 5 years across different drug modalities. Next slide. We support 4 core services: pharmacokinetics, immunogenicity, biomarkers, and CMC analytical. Next slide. And the way that we do it is from a variety of different platforms. So today we're going to be highlighting a PK LCMS, cross-validation, and assay transfer. We will be talking about assay transfers for cell-based assays such as flow cytometry and in the CMC analytical space, when we will also be covering immunoassays such as anti-drug antibody assays and the different considerations for how do you actually go about cross-validating an assay. Next slide. So what is assay cross-validation? Well, cross-validation of assays is required when multiple labs are running the same assay, and you need to demonstrate how the reported data is related within a single study. How does the data relate from lab 1 to lab 2? As we think about the current global drug development landscape, it is often required to run assays in multiple labs due to timeline constraints, sample shipment restrictions, and other factors. Due to the complexity of having multiple labs running the same assay, there's a number of challenges that need to be overcome, such as communication, whether it's lab to lab within the same company or across companies, dealing with the same assay validated in multiple labs, changes over time in assay reagents, matrices, and procedure, changes in environment, subtle differences in procedure and instruments, and reagent or sample shipment issues. So now that we have an idea of the complexity, how do you ensure a successful cross-validation? Well, in today's seminar, we're going to share different strategies and case studies that highlight the different ways to overcome these cross-validation challenges across a variety of different assay types. So the first assay type that we're going to cover today is a PK assay that was supported on the LCMS platform, and Pavan Pratipati, next slide, is going to be sharing his experience. So Pavan, please take it away. Pavan, you are muted.
Yeah, thanks, Lynn. Sorry about that. For this case study, I will walk through a small molecule prodrug and its active form, which we will refer to as drug A. Next slide, please. The original LCMS MS method was developed and validated by the originator lab, which also analyzed the initial samples. After the method was transferred to bioanalytics, we performed partial validation for both compounds. This included selectivity, matrix effect, carryover, intraday accuracy, precision, reinjection integrity, and working internal standard solution stability. The table on the right summarizes the partial validation data for drug A and shows that the method performed well in our lab. So the next question becomes, how comparable are the results between laboratories? Next slide, please. Historically, under 2018 bioanalytical method validation guidance, cross-validation was typically evaluated using QCs and study samples with acceptance criteria similar to ISR. In practice, that meant at least 67% of samples were expected to be within plus or minus 20% of the original value. ICH M10 now recommends using low, mid, and high QC levels along with study samples when available, ideally at least 30 samples to determine interlaboratory reliability. To evaluate bias, tools such as Deming regression, Bland-Altman analysis, or other appropriate statistical approaches can be used. Another option is to compare concentration versus time curves for study samples. That said, the guidance does not define specific acceptance criteria for cross-validation. So the interpretation requires scientific judgment. Next slide, please. We perform cross-validation using QCs in triplicate at low, mid, and high QC levels. Those QCs were analyzed by the originator lab and then provided to us along with more than 30 blinded study samples. The figure on the right shows the comparison of study sample concentration from originator lab and bioanalytics. At a visual level, most of the results appear to align reasonably well between both the labs. To look more closely at potential assay bias, we evaluated data using both Bland-Altman analysis and Deming regression. Next slide, please. Here, we are looking at the Deming regression plot comparing study sample data from both laboratories. Common expectations for Deming regression include slope within about 0.9 to 1.1, or sometimes 0.95 to 1.05, depending on the context. We also typically look at 95% confidence interval of the slope to include 1, and the 95% confidence interval of the intercept to include 0. In this example, the slope was 1.05 with the confidence interval of 1.02 to 1.1. Because 1 was not included in the confidence interval, the data do not fully support null hypothesis, which suggests that proportional bias may exist. To further explore the potential bias, we also used a Bland-Altman plot to evaluate agreement between both laboratories. Next slide, please. Bland-Altman plot gives another way to understand agreement between two measurements. It helps quantify both size of the differences and how those differences are distributed across concentration range. Ideally, we want the mean to be as close as to 0 and the bias to be randomly distributed, and no evidence of proportional bias and relatively narrowed 95% limits of agreement. On this plot, the x-axis represents average of the two measurements and the y-axis represents the percent difference between them. The solid red line shows the mean difference, while the dotted lines show 95% confidence interval around that mean. The shaded area represents 95% of limits of agreement. In this case, lower concentrations do not appear to be randomly distributed as the higher concentrations does, which suggests bias might exist at the lower concentration range. Next slide, please. So the main takeaway here is that current regulatory guidance does not provide a defined acceptance criteria for cross-validation in the same way it does for ISR. As a result, drug developers need to make scientifically justified decisions based on the totality of the data. Whenever possible, cross-validation should include both QCs and actual study samples that span assay range. If study samples are not available, additional QCs across the range can still provide useful information about assay concordance between laboratories. Statistical tools such as Bland-Altman analysis and Deming regression can be very helpful for evaluating potential bias. It is also important to understand what each statistical tool tells us. Bland-Altman focuses on the agreement and the differences, while Deming regression evaluates the relationship between two data sets without assuming that either lab is a true reference. That brings me to the end of this case study. Thank you, and I look forward to discussing any questions with the panel at the end of the webinar.
Thank you so much, Pavan. As Pavan said, feel free to put any questions for him in the chat. Our next case study focuses around the transfer of a replication-competent lentivirus assay by qPCR and will be presented by Brian Gulick.
I'm going to talk just briefly about an assay transfer that we did from one of our labs to another in the PCR space. So this is a safety assay, commonly referred to as an RCL assay, that we have set up in a number of our labs in order to test the VSVG sequence from lentivirus. This is part of the envelope to show this functional molecule would be in existence in a given sample. Next slide. So when we were looking at transferring this assay from lab 1 to lab 2, we basically wanted to look at as much homology as we could have. So we have very, very similar PCR equipment. We were using the exact same sequences with the same fluorophores for both of our primer probe sets in both labs. So these were TaqMan-based assays. We were using the same extraction systems, both being the Kingfisher Flex, a magnetic bead-based extraction system, utilizing the same extraction kits as well, and then the same reference material was used in both assays. Important to note, different from the first presentation you saw, that we were not cross-validating this assay, but transferring it to have it stood up in 2 separate labs. Next slide. And so what you can see here without a lot of detail is we set up the assay with the same calibration curve range. So from 10 copies to 1 million copies per well. We demonstrated consistency in linearity across both assays. We kept the same number of thermocycles during the qPCR reaction conditions. So both were set at 45 total cycles. During the independent validations, we did see slightly different limits of detection. This is not uncommon because that's a quite variable range, well below the LLOQ of the assay, but even then quite comparable between 2 and 4 copies. Overall, we kept the same acceptance criteria for accuracy and precision. In this case, it was 25% at the upper ranges, and then due to the higher variability that we routinely see at the lowest limits, we had an expanded criteria of 45% at the LQC and LLOQ. We demonstrated consistency between both of these sets in all of our accuracy and precision runs, with the overall being within that 25 or 45%. Again, selectivity was demonstrated comparably. We looked at some additional stability as well at both sites and showed comparable performance there. Next slide. And so this is a busy slide with a lot of numbers on it, but basically just showing a summary of one of the accuracy and precision sets. So we have 3 runs of accuracy and precision across the full range of the assay. And I think the kind of important points to highlight are at the very bottom, where the interassay CV and RE were both well within the determined criteria, previously determined criteria for these assays. Let's see, next slide, so we don't need to belabor the data too much. So really just a brief summary. I know this one was quick. We actually, I think, set up for success with having very, very comparable, basically everything between the 2 labs. We transferred the method and all of the method details, utilizing as much of the same equipment and processes as possible. And what we saw was a very easy and consistent performance between both labs. In the second lab, we were additionally able to add another matrix to the list of validated matrices. And so we can support both PBMCs in both labs and then whole blood in lab 2 as well as the PBMCs. Ultimately, it was a very straightforward process, and I think you typically will see that when you can utilize as much homology between labs as possible. Next slide. I think that's all that I have. Thank you.
Thank you so much, Brian. And any questions you have for Brian, please feel free to put them into the chat. Our next case study focuses around the transfer and cross-qualification of a flow cytometry-based assay, and Michael Mullen will be taking us through that. Michael, hello.
Hello. Next slide, please. 360 Biolabs, our Australian site, developed and validated a 13-color flow cytometric immunophenotyping panel. This panel is to measure the effect of our sponsor's drug targeting KLRG1 on cytotoxic T cells. In particular, the sponsor wanted accurate cell counts of specific cell populations. The matrix for this assay, however, is whole blood with a stability of roughly 24 hours. As the clinical trial was moving to the US, our job was to transfer the assay from the Australia site to our Boston site. In Australia, the team validated the assay on a BD LSR4 Tessa. However, our Boston site is equipped with Beckman Coulter Cytoflex LXs. Our sponsor was also interested in adding a B cell marker, so we added a CD20 stain to the panel. We discovered some issues with the current staining panel moving to a different cytometer system with different lasers and detectors. There was some spectral overlap that was not able to be rectified with compensation. So prior to sample testing, we had to go back into panel design optimization. Next slide, please. The original assay validated was a 13-color panel. As I said, the sponsor requested the addition of a B cell marker, so we added a CD20 stain. This slide, we're looking at a depiction of the laser and detector systems of the Beckman Coulter Cytoflex LX. Above the yellow-green laser, the second from the left, you'll see a circled red box that's CD20, and we initially conjugated it to PE-Psi5 fluorophore. This fluorophore is primarily excited by the yellow-green laser on the cytoflex, and the nearest fluorophore also excited by the yellow-green laser was PE. Both, however, utilize a different detector, and populations were distinguishable. The issue we quickly discovered was that PE-Psi5 was also being excited by the blue laser, as you see over in the middle of this depiction here. And PE-Psi5 was being detected in the PerCP-Psi55 detector, which we had conjugated to CD3, our primary T cell marker. This spillover was unable to be compensated as there was a large spectral overlap. Next slide, please. As you can see in the spectral viewer here to the left, both these fluorophores are emitting strongly in the 665 to 715 nanometer detection range. This moderate to high spillover of PE-Psi5 into the PerCP-Psi55 channel is leading to a false cell population. If you take a look to this flow plot on the right here, it should be a bivariate flow plot with no double positive. There should be a B cell population at the top left and a T cell population at the bottom right. It's biologically impossible to be both a B and T cell, so this population in the red box we're seeing to the top right is biologically impossible, indicating a staining compensation issue with our panel. So we went back into panel optimization and researched for better fluorophore pairing on the cytoflex. Next slide, please. We decided to move CD20 from PE-Psi5 to being conjugated to ECD. You can see this in the bottom detector setup there. The leftmost red box, CD20 ECD. This had a detector specific for this color, as ECD is a proprietary color of Beckman Coulter. Also excited by the blue laser was Alexa Fluor 488. However, that utilized a different detector and the populations were distinct and easily gateable. CD3, which was initially on PerCP-Psi55, we moved to Brilliant Violet 650 to ensure that it was excited by a different laser than ECD. The spectral viewer to the top left shows that now these two colors have distinct emission peaks. In the flow plot to the right, you can see what the bivariate plot should have looked like before. There's a T and B cell populations that are now easily definable, and there's no false double positive T and B cell population. Next slide, please. As I said earlier, our sponsor's primary goal was accurate cell counts of specific cell populations. So absolute cell counting was necessary. The first method we tried was Beckman-Coulter's recommended method, fluorophore bead staining. This would have been beneficial as it would allow us to maintain assay protocol with washing and centrifugation steps, as the fluorophore counting beads would then be added to the stain sample at the end of the prep protocol. The benefit of this would be the addition of a live dead stain, as live dead staining requires washing and centrifugation steps. However, these beads and our cell populations of interest demonstrated very similar forward side scatter characteristics. As you can see in the top flow plots here, the second plot after the singlets gate, the fluorophore beads are this orange population. And you can see they're not easily defined. They're very close to the cell populations of interest. So gating was subjective and not consistent between samples of the same patient, never mind between different patients. These beads are artificially affecting our cell populations, leading to inaccurate, inconsistent cell counting. So we tried a different method, the industry standard true count tubes from BD. These tubes have a known number of beads in each one. And as you can see in the flow plots to the bottom here, the beads and our cells were easily defined populations. This top left plot of the blue populations, you can see the orange gate here is our cells and the pink gate above it is the beads. Gating was clear and straightforward and consistent through patient time points and between patients. The only limitation with this method is that it required a lice no wash method, so we were unable to implement live dead staining. However, cell counting was accurate, so our sponsor was content as their needs were met. Next slide, please. This slide here is just a quick walkthrough of how these counting beads work. We're able to ensure that we're counting accurately every run. The table on the left shows you how percent recovery is analyzed. There's a known bead count provided on the COA by BD, and we know the volume of sample, staining mix, and buffer that's added to the tube, so we can calculate our expected concentration. The cytometer reports how much volume of sample is processed and how many beads were counted, our observed bead concentration, and we can calculate our recovery from that. Using our bead count along with our parent population statistics, we're then able to numerate cell populations to support our sponsor's needs. Next slide, please. Bioanalytics successfully transferred a 14-color flow cytometric panel from Australia to Boston. Bioanalytics Boston has now supported over 250 patient samples through this clinical trial. Although it wasn't a straightforward transfer, the team overcame all the obstacles. We learned that optimization was required for cross-validation of this flow cytometric assay between platforms, and that it's often difficult when switching panels to a cytometer with different laser detector setup. It's important to be cognizant of tandem dyes and their potential spillover, and most importantly, to demonstrate comparability between platforms and altered panels. This cross-validation highlighted Bioanalytics' ability to overcome technical troubleshooting challenges by thoughtful experimentation and assay optimization. Thank you all for listening.
Thank you, Michael. Really appreciate that. Feel free, remember audience, to put any questions for Michael into the chat and we'll answer it at the end of this webinar. Our next case study focuses really around the GMP space, where Jessica Weaver will be presenting the multi-site co-validation of a cytotoxicity potency assay using different plate readers and critical reagents. Jessica, please take it away.
Thank you, Lynn. When transferring an established CMC bioassay, it's far more complex than a simple scientific handover. It's a formal regulated process designed to prove that the receiving laboratory can perform the method with the exact same accuracy and reliability as the originating lab. Some of the documents that we follow are ICH-Q2R2 specifically. That is a global document accepted globally within the US and EU. Within the US, we also follow USP 1224, and within Europe, the EU GMP Chapter 6 for quality control. These guidelines tell us what the authorities are expecting out of a transfer, but the operational challenge is the how. So as we step into the operational challenge of this case study, let me walk you through the schematic of the mechanism of action. This is an antibody drug conjugate, and as and the antibody drug conjugate is a monoclonal antibody that is linked to a cytotoxic drug. The antibody binds to a specific surface receptor on the surface of the cells, and then it is internalized within the cells. Once internalized, the antibody is cleaved, and the toxin then has a cytotoxic effect on the cells, causing cell death. Next slide, please. Breaking down the assay procedure, the first step was to perform a dilution series of the drug material. Target cells are then added at a known density. The plate is incubated for 3 to 4 days in the standard incubation conditions. After the incubation, a detection reagent is added, and the data is read on an absorbance plate reader. The data is analyzed using a nonlinear regression where a 4-parameter curve was fit to both the reference material and the sample material. Comparisons of those dose response curves for the reference in the sample allow determination of a relative biological activity of the samples. One interesting point to note for this assay is that while preparing for the formal transfer, there was in-depth conversations with the analysts at the originating site and our site here in Durham. Some of the critical steps around cell culture handling, cell culture counting, as well as very particular pipetting practices were noted. Upon our request, the originating lab provided videos of the assay execution to aid in the training for the transfer, and that was such an added benefit that those videos are still used to this day for training on the assay for new analysts. Next slide, please. As discussed, the drug is an ADC, and after setting transfer goals and assessing risk and performing gap analysis, we knew the following. The drug is already validated at a phase 3 validation at the primary site. The risk assessment determined that the multi-site co-validation was needed due to the current status of the validation. Bioanalytics, in this case, served as a secondary lab for the co-validation, and we also identified reagent FBS was unable to be imported into the US. Another thing that was identified during the risk assessment was the difference in plate readers at bioanalytics versus the plate readers and the analysis software at the originating lab. Next slide, please. So let's dive a little deeper into that gap analysis. So the plate readers at bioanalytics at the time were the Biotech Neo 2s, while the originating lab had the SpectraMax. So there were concerns whether or not the impacts would impact the co-validation assessment using 2 different plate readers. So one of the notable differences are some slight differences in the detection of the machines. The photomultiplier tubes are slightly variant. So one of the things that was carefully investigated and tested was seeing that while there were some shifting in the readout values of the data using the different plate readers, it did not overall impact the relative potency results. So therefore, both plate readers were deemed acceptable to use within the assay validation. The differences in the software were evaluated as well, and the data from each plate reader was ultimately put into SoftMax Pro for analysis, mainly because of comparison to historical data. So the primary test site was also in Europe, and the assay was developed in Europe. The FBS there that they were using is unable to come into the US due to FMDA vaccination requirements that the USDA has here in the US. So we took a very careful and equivalent source of FBS, looked at that in the cell culture, looked at that in the growth history of the cell culture before performing any of the transfer activities. Next slide, please. During the risk assessment and finalization of the transfer protocol, the parameters and acceptance criteria were determined. We looked at linearity across 5 different target concentrations with 3 independent replicates at each target concentration across 2 analysts. Precision reducibility were also investigated. Accuracy was investigated as well. And we also looked at robustness with force degraded samples for the indication of stability. As you can see here on this particular slide, there was clearly defined criteria in place for each of the parameters we tested in the assay. It is important to note that independent replicates were an important factor, especially for a transfer assay. We determined it'd be better to get a look at true variation within the assay, both inter and intra-laboratory variations. So we looked at across plate, across assay plates, across assay test days, across labs, across analysts, with those true independent replicates. Next slide, please. So the results are shown here for the bioanalytics testing specifically. Overall, this assay transfer was successful, and the co-validation met the predefined acceptance criteria for all labs that were involved. The data confirmed that the assay performed as expected across sites. One important note is we also looked at, in addition to the acceptance criteria outlined in the slide before, we also assessed the look for bias between the different sites and systemic error. So for example, if one site had results that were trending low, even if they were within specification and they were still trending low, it's important to note this, especially with different variables like new instruments, new analysts, new reagents. You could have a bias due to one of those variables that you don't necessarily see if you're not looking specifically at the bias. So in this case, we did not see any bias within those variables from the instruments, the analysts, the labs, the reagents. So that was great, and everything went really well, and we had a successful validation, and they're currently supporting late phase stability studies for lot release. With that, Lynn, back to you.
Thank you so much, Jessica. Really appreciate that. Remember to put any questions for Jessica into the chat. And so now we're going to finish off with a discussion around best practices for ADA cross-validation in different statistical considerations. And I've seen some questions in the chat around this, so I'm really looking forward to Todd's discussion. Todd, can you hear me?
I can hear you. Can you hear me, Lynn?
Yes, I can, Todd. Take it away.
All right, thanks. Yeah, we'll be talking about immunogenicity assays here, and in scope are both ADA assays and NAB assays, so anti-drug antibodies and neutralizing antibodies assays meant to detect and measure an immune response to the dosed therapeutic. And in these scenarios, really, we have a difference in these assays between, say, the PK assays, where we have a nominal reference standard that we can measure and then quantify against. In these cases, these assays are typically qualitative in nature. And so based on that nature of the assay, we typically set up thresholds or cut points that are based in the variability of the assay, whether that's analytical variability or biological variability. And we characterize that variability and then use it to establish thresholds, cut points that call positive or negative, and then we go on to assess parameters relative to those cut points, say sensitivity of the assay, so how low of an immune response can we detect, or the drug tolerance or target tolerance, how much free of residual drug or residual target in the samples that we're measuring can the test resist or be robust against. And so there are various factors that contribute to differences in assay performance when we're talking about methods that might exist, two qualitative methods in two different laboratories. From the capture and detection reagents that we use, if we have more label, it might result in a absolute difference in magnitude of signal at one lab versus another, or our negative control pool that we use to normalize samples against. If two labs are using two different pools, that might set up differences and normalize signal. The drug lot that we're using in the confirmatory assay might have differences that drive a different inhibition between the two labs. And so these differences, may exist, but really the point of the cross-validation exercise is to characterize those differences and ultimately speak to the impact. So in the cross-validation exercise, where we have two different methods validated at each of two labs, they might have two different cut points or two different sensitivities or two different drug tolerances. And unfortunately, there's not a statistical test that we can use to say, is the sensitivity in this lab the same as the sensitivity in another lab? There just is not a statistical test to say, are these two numbers the same? And so we know that, feeding into, say, the cut points and then the characterization against the cut points of these assays, there's various practices that lead into the cut points, say outlier identification and discrimination, and differences in practices, equipment, and acceptance criteria across the labs and across the methods. And these set up challenges, but also opportunities to start comparing and ultimately looking at where potential differences might stem from. And so as we look at the comparability of methods, we might start looking at positivity rates between the different methods or between the labs. We're looking at differences ultimately in titer dilutions across the labs. And so we have these different data sets that we might pull from and then apply different statistical methods. Next slide, please. So taking a step back and kind of higher level picture, I think, immunogenicity is the qualitative nature relative to quantitative assays. I'd say in the quantitative assay realm, the practice around cross-validation is probably a bit more mature. If we look at the guidance, we have that reference in M10 that starts to call out statistical tests and really call for critical thought to be put into comparing these different data sets. When we look at the regulatory expectations and the recommendations, consensus recommendations in literature in the immunogenicity assay realm, I'd say it's less mature than the practice and the recommendations issued so far in industry for PK quantitative methods. And so that said, we do have some references going back to the 2019 FDA immunogenicity guidance, and there they talk about reproducibility and cross-validation. Those terms are used kind of whimsically or interchangeably, at least in those that guidance. But it's general vague guidelines, and then we move on in time to the 2023, the APS harmonization paper, Myler et al. 23, and in that paper we outlined pretty in depth some different practices around how to compare validation data from two different labs or two different methods, and how to start looking at that relationship between those data sets. As we were shopping that paper around to different conferences after its publication, some members from the FDA actually brought forth some expectations and said, hey, why don't you go back to the AAPS harmonization paper on ADA assays and add in some of the practices and recommendations that were in the NAB assay harmonization paper. Why don't you introduce those into the ADA harmonization paper? And so earlier this year, there was an addendum to the Myler et al. White paper on ADA harmonization, and brought forth some of the same recommendations that were issued for the NAB harmonization into the ADA harmonization. And so that paper introduces some of the experiments that we can do to generate data sets to go about the comparability exercise in a similar way across now ADA and NAB. Next slide, please. So what can we do? Similarly to PK, we can take a panel of patient samples, say 30, say 50. I don't think there's one magic number, but a sample size of significant, not trivial number, and we can analyze those samples across the two methods or across the two labs. Those samples are not always available, and so in lieu of having patient samples, we see different reasons for why samples might not be available. They just might not have been tested yet in the second lab. There might be limited volume. There might be freeze thaws already exhausted in the samples that we do have. They might be in a rare population where volume's limiting. But for whatever reason, if we don't have samples available to analyze in both methods, we can spike the controls. We might spike in a low PC and a high PC and include a negative control and analyze those samples at two different labs. Thought should be put into the specific assay at hand, and if we have domain specificity tiers, to include domain characterization, but to test the relevant controls for the assay, and to generate data sets, and then we can start comparing the data. We could compare the raw data, the raw response, whether that's ECL or ELISA, OD, absorbance. We can compare the normalized signal to noise negative control ratios. We could compare the titers for positive samples. And I think it's important to look at probably all of these different data types, because if we're looking at just raw response, we might miss something that an impact maybe that becomes apparent when you normalize the signal. And similarly, if we're just looking at the normalized ratio, we might miss maybe differences that might appear or be present if we were just to consider the raw response. I think there are case studies out there where if we don't, raw response and normalized signal might look good, but if you actually look at the titration, there might be some differences there in the tighter curves. And so I think it's important to take a 360 look and to compare, as many data types as are available for any given assay to get a 360 look to make sure we're doing our due diligence and covering our bases and looking at all the assay output and not just a myopic view. Let's go to the next slide. So yeah, I think in approaching this exercise, it's important to understand that big picture context, and the purpose of the exercise is really to understand the extent of any differences in assay performance, and to put into context the impact on interpretation of clinical sample results. And so ultimately we'll have a data set of sample results from one lab or one method and another lab and another method, and those are getting combined to look at incidence across a population level. And we have to keep that end exercise, that end purpose in mind, and to look at any differences that may appear as we start comparing raw signals to how it will impact interpretation of the clinical immunogenicity. I think I would caution against applying predefined acceptance criteria. The exercise is not meant to, be a checkbox and to say, okay, to 80% here, 80% here, check, call it good. I think the exercise calls for an in-depth analysis from a 360-degree angle, and then a meaningful interpretation of any differences that may start to emerge as you walk through the analyses. What exercises can we do? We can start to overlay, distributions of those different signals, that raw signal, the normalized signal. We can apply t-tests, and we can do, even rating tests, looking at the positive and negative. You can count up, the positives in one lab and the second lab and the negatives, and look at, say, the Cohen's kappa statistic, which is a rating assessment of the agreeability between qualitative data. We can get a quantitative output and assess that coefficient against a level of agreement based on where that coefficient lies. But I think there's, again, going back to that acceptance criteria and whether we should have checkbox criteria or whether we should really be thinking about the differences we might see, and then how that gets put into context as we interpret clinical sample results. Ultimately, that's the exercise. And so, even boiling down to a Cohen's kappa coefficient and calling it good based on a range, I think there's need to actually think about the data that we have to make sure we end up at a fit for purpose approach. Then the last thing I'd say within the immunogenicity space, I think the integrated summary of immunogenicity is really an excellent forum by which you can tell the story of the immunogenicity of a given program. And as reviewers, regulators pick up a BLA submission, say, and start to understand the complexity that often exists across a development program in methodological changes and site changes. The underlying labs and methods can be a complex story to tell. And so the integrated summary of immunogenicity is really an opportunity to tell that story of the assay life cycle, to explain the rationale for the evolution in methods and across labs. I think regulators do understand that these methods do change over time, oftentimes are improving over the course of the development of a program. And so the integrated summary of immunogenicity can act as a bridge connecting the bioanalytical methodology with the clinical immunogenicity evaluation. And that's a apt forum to tell the story, to include these comparisons as from the cross-validation exercises, to tell that complete picture and hopefully at the end of the day, reducing the number of questions that arise during the BLA review process.
Thank you so much, Todd. And so I think just a few quick takeaways before we open the floor for Q&A. Bioanalytical methods such as PK, ADA, PD, and biomarkers, and CMC potency are a key component of clinical studies, and ensuring interlaboratory concordance is essential to maintain data quality in alignment with regulatory expectations. Robust assay cross-validation requires demonstration of equivalence in key acceptance criteria. Variability introduced by site-specific reagent lots, instrumentation, and analyst performance can all introduce bias. Assay transfer examples reviewed in today's presentation have outlined practical strategies to overcome the challenges associated with cross-validation. And as Todd was just discussing, analysis of assay bias or concordance can utilize a number of different statistical approaches, but the interpretation of bias should be evaluated within the context of the individual program and guided by fit-for-purpose scientific rationale. So with that, I invite all of the other presenters to join me right now as we open the floor to Q&A. So Todd, I think you kind of spoke to this at the beginning, but I think this was also addressed specifically to Pavan for his PK case study in terms of how many clinical or commercial samples do you recommend to test for inter-lab assay performance? So first, Pavan, what has been your experience? And then Todd, from a statistical perspective, what would your recommendations be?
Yeah, per guidance ICH M10, it clearly specifies 30 or more.
Mm-hmm.
Yeah, even for statistical significance, the greater number is better, but we always don't have samples of that size.
Right, right. Todd, any input from your end from the statistical analysis component?
I would just say, I think that, that 30 or more number comes from, as you look at the relationship of sample size with standard deviation, as your sample size increases, we know that standard deviations drop. And so, they drop a lot in that first, sample size of 0 to 10, 10 to 20, as you get up to 30 to 50, that's where your standard deviation levels off. I think that's the reason for some of these numbers. It's the reason, in a cut point exercise, we typically include 50 individuals because that's, but I think, beyond just a single number, I think you do have to look at your entire data set and your method and what you know about the method. And if you have, say, a proportional bias, like Pavan was showing earlier, maybe you include, samples from 2 different regions where you see different levels of bias. Or in the immunogenicity space, a question that comes up a lot is, should we choose all 30, positive samples, or should we include 15 positive and 15 kind of negative, or that region in between where, we would expect analytical variability to exist, and so samples might be flipping from positive to negative. These are the details that I think as you approach any given exercise, you need to think about your assay and the context of use of the assay, and then go about designing a given assay transferend or cross-validation exercise with an informed number that makes sense for your context of use.
Thank you, Todd. And, very much a related question, but this time for the qPCR platform. Brian, there is a question on cross-validation of PCR methods. Do you need to test both QCs and 30 incurred samples and assess if there's any bias like LCMS or a ligand binding assay? Is there the potential for bias across this platform as well?
Yeah, I mean, I think the question is very valid, and I think it makes a lot of sense if your intention is to actually cross-validate. If you're intending to be testing samples from the same studies across multiple sites, you definitely want to ensure that comparability. In the case of what I presented briefly on, we intended to fully separate those studies. There were studies that were being done more regionally, and there wasn't the specific need to cross-validate. But I think it's something that makes sense. You definitely want to be sure that you don't have bias in your assays. One thing to note also with an RCL assay is it should be a negative, and so testing tons and tons of samples that all say 0 are maybe not as worthwhile. I think testing QCs to ensure consistency in the assay probably makes a lot of sense if you intend to test in multiple sites.
Thank you, Brian. And I think we have time for one more question, which is really, for a PK assay transfer, either Bland-Altman plots or Deming regression, are they both required, or should it be only introduced when bias is obviously observed? Todd, from your data science perspective, what do you think about this?
I mean, I actually start from the regulatory perspective, and I would say neither of them are required. I mean, I think what's required is to think about your data set, to bring critical thought to the two different data sets, methods, labs, whatever is feeding into the need to compare. And then yes, Bland-Altman, Deming regression are called out in M10. That's the first time that they've had specific examples of tests, analyses to use in the regulatory expectations. But also, so were removed acceptance criteria from M10. And so the reason for removing the acceptance criteria was to promote the notion that there can be bias and that's okay, but we need to understand what the bias is, describe it, and then be able to incorporate that into a downstream interpretation as we look at population level PK, PD, immunogenicity, whatever the exercises at hand. We've seen slightly different, depending on the data sets. I think Deming and Bland-Altman can, be aligned or show slightly different nuanced views of the data. As Pavan presented, there's an assumption that one assay or one of the methods is kind of the gold platinum and standard in one of those tests, and the other does not work under that presumption. And so from how those tests and visualizations work, I think there's differences. I wouldn't go to one or the other. Typically, we're looking at both, and maybe we're moving forward with one as kind of the best way to look at the data for that given program. But again, I think thought around the data sets that we have and the analyses of the tools that we're using is really the most important thing to ensure that we arrive at appropriate conclusions for any given exercise.
Thank you so much, Todd, and I couldn't agree with you more. Really just thinking critically about the science that you're doing. And with that, we are brought to time, so I'm going to transfer it back to Corey.
Excellent. Thank you so much, Lynn, and thank you to everyone. A really insightful Q&A session, and thank you to our audience members for submitting some questions. We had a few other questions that we didn't have a chance to get to, but rest assured they'll be passed over to the team at Bioanalytics for further follow-up. If you do have any other questions, though, please feel free to reach out to Lynn. Email address is up on screen here, and I'm sure she'll be happy to chat with you after we go off the air today. I will thank everyone once again for participating. I've added a link in the chat box where you'll be able to view the recording of this event, and you can of course share this link with your colleagues so that they may register to view it as well. You will also be receiving a follow-up email from XTalks that will give you access to the recording, and after we wrap things up here, a survey window will appear on your screen. Your participation in this survey is always appreciated as it helps us to improve our webinars. Now, before we sign off, I did just want to take one more moment to thank you, Lynn, thank you, Jessica, thank you, Pavan, thank you, Brian, Michael, Todd. Really appreciate your time here today. Hope our audience found it insightful and informative, and we wish you all a great rest of your day. Take care, everyone.